All right, we're going to go ahead and get started. Thank you everyone for being here. There are some repeat faces from last night as well. So thank you everyone for joining us for our research seminar today. This is a hybrid event, so welcome to the folks that are online as well. For those folks that are online, if you have any technical issues, you can go ahead and put your questions into the chat box, and our volunteer Roseanne will help you address those. Also, if you have any questions for our speaker, you can use that same chat box, and she will read those questions out to us at the end of today's presentation, and we can get those questions answered. For those folks in the room, most of you have done this with me before, but remember, if you have questions, we're going to hold those to the end of today's talk as well. Raise your hand and I'll bring you a mic, or you can use the mic stand that's up there on the side. If you haven't already, can you also make sure you sign in for us? I say this every week, but this is how we are able to buy cookies and coffee. We need to justify that I am not eating them all. So if you can sign in for us, that would be great. A couple of really quick announcements before we get started today. I wanted to let everyone know that next week's seminar, we have Maria Winters from the University of California, Los Angeles. The Coastal Flood Lab will be here talking about the observations and modeling of anthropogenic dune evolution and erosion in Southern California. So that will be an interesting talk for us. And that kind of plays with this month's Science on Tap, where we have staff members from DLCD coming to talk to us about changing Oregon beaches and dunes and the land use planning goal 19 that is effectively used to manage those spaces. So if you're into those kind of things, we've got a good month of things coming up. So come join us for those. And as always, if you are interested in any of marine science, look at me, field marine science type events, you can go to our webpage and log into the events page and you get all the information you need there. But for today, again, we are so excited to have this year's Liver and Weber visiting scientists with us to present. This particular fellowship honors Dr. Liver and Weber, who is the director of HMSC from 1977 to 2002. And this fund helps support expert researchers coming and staying at HMSC to build the foundations of long-term collaboration with the HMSC community. If you are interested in hosting a Liver and Weber visiting scientist, you can do like Lee did last fall. Reach out to me and I can give you all that information and we can get your speaker here as well. But as I said, Lee was the one that invited this year's speaker. And so I'm going to actually hand this off to Lee to introduce Dr. Reed.
Thanks, Cinnamon. Okay, here we are again. For those of you who don't know, I introduced Andy last night for his Science on Tap. That was excellent and recorded, so you too can catch up on everything. But I am honored to host Andy here. For those of you who don't know, Andy was my graduate advisor at the Duke Marine Lab for both my master's and my PhD, which scary for him as well as me, I just figured out was 25 years ago. I know, when we first met. And so really, you know, last night I mentioned about how much I learned from him in those formative years. And he really is a brilliant ecologist and scientist and mentor and conservationist. But it's been great having him here again, because we really haven't been able to connect very much except at random conferences here and there since I graduated many years ago now also. But this has been amazing just to learn again from him in all these conversations we've been having, his presentations, seeing my students and other people in the Hatfield community also learn from his wisdom and knowledge. So that's amazing. So I'm grateful for the opportunity to bring him here. But yeah, so who is he? So for those of us who don't know, so Andy is truly a preeminent leader in the field of marine mammal science and conservation. So he's globally recognized for a lot of his work, pushing the envelope on how we study and how we interpret the data for both understanding their ecology of cetaceans, but also their management. So he's served on various committees at both national and international levels, including in the IUCN, the IWC, take reduction teams, as well as serving as the president of the Society for Marine Mammalogy. And currently he was nominated last year by President Biden to be a commissioner for the U.S. Marine Mammal Commission, which provides oversight for marine mammal policies and programs to be carried out at the federal level. So it is really special for him to be here and share his insight into that world as well. So with that, I think I'll hand it off to him. I'm so excited to hear about beaked whales and his viewpoints on what they're learning.
Thank you, Lee. I guess it has been that long ago because when Lee and I first met, we played soccer together and I could keep up. And that's no longer the case. I will say that as one progressed through one's career, at least for me, the thing that I am proudest of is the accomplishments of my students. And I'm so proud of what you've done here. It's been amazing to meet with your lab. And it's just been, my head's kind of exploding. So if that happens during the talk and my head explodes, you can just leave. So I'm going to do something a little different today, and I hope it works. And if it doesn't work, you'll have to forgive me. But I'm going to take a different spin on the work that we do. And we're funded, as I'll explain, by the Navy to do controlled exposure experiments with this unbelievably strange and wonderful beast, Ziphius cavirostris. But I'm not really going to talk about that very much other than to frame the things that I think are really interesting, which are the fundamental aspects of the behavior and ecology of this strange animal. So let's see how that works. And I want to give a shout out to Annie Harshberger, who a couple of you know quite well. When I was thinking about giving this presentation, I was thinking about a catchy title. And Annie, who's a PhD student in my lab, had just come back from Denmark, meeting with one of her committee members. And she said, I can't believe it. In Denmark, PhDs just last for 3 years. And Annie's in the end of her 3rd year. And she said, that's unfathomable. And I'm like, I'm taking that and using that in my title. So okay. So we're going to talk about this strange and wonderful creature. And just a word at the beginning, most people refer to them as Cuvier's beaked whales to honor this individual, Georges Cuvier. And some of my students have asked me very pointedly not to no longer refer to it as Cuvier's beaked whale because of some of the negative aspects of Cuvier's life. And I'm not going to go into it today, but if you Google the name Sarah Bartman and look at the interaction between Cuvier and Sarah Bartman, racism, misogyny, all kinds of things. And I think it's not appropriate for us to honor him by naming this incredible species and referring to his name when we do that. So I'll call it goosebeak whale, or what I like to do is just call it Ziphius, because Ziphius feels right for this species. So I'm always hesitant to put a phylogeny up when Scott Baker's in the room, but so we can argue about this later, but I just want to make this as Michael McGowan from a Michael McGowan paper. All I want to say is that beaked whales, and Scott would, I think, agree with this, are a diverse and very poorly studied clade, maybe the second most diverse clade of cetaceans, right? And we know virtually nothing about them. We know more than we did 20 years ago, but still we're discovering new species thanks to the work of people like Scott and his lab. And it's just, they're just, to me, unimaginably strange animals and wonderful animals. And so my whole career, I really didn't work on them very much. I worked on coastal animals, and then I've worked on a bunch of different delphinids. And it's been really extraordinary to get to know the strangeness of the beaked whales a little bit. Ziphius is probably the most cosmopolitan of the beaked whales, one of the better known, and maybe one of the more accessible ones, I'll show you. If I was here 20 years ago, and I know that's a long time to some of you, it's not so long to me or Lee. But if I was here talking about Ziphius 20 years ago, I would be talking about insights and inferences from strandings. And that's about all we knew. We knew some things about the animal, but nobody, if somebody had said, I'm going to study live Ziphius, people would have just laughed at you. It wasn't possible. And then this happened in 2000, or this report came out in 2000 after an event in the Bahamas in which a US Navy training exercise resulted in a mass stranding of cetaceans, 17 cetaceans, including 9 Ziphius cavirostris. And the report from the investigation of the causes of that stranding event that was co-authored by both the Navy and NOAA concluded that the mid-frequency active tactical sonars that were used during that exercise were most likely the factor that led to the stranding of these animals. And that just caused a whole furor. And if you weren't alive at that point, or you were too young to take notice, it's hard to know exactly how much things bubbled up as a result of that. But it led to books like War of the Whales, Sonic Sea. There was a whole, as I said, furor about what had happened. The Navy really didn't handle it very well in the immediate aftermath of that stranding. And some folks in the Navy were concerned that every time these sonars were being used, and they're used to find typically quiet submarines, and they're used by most Navy vessels, they imagined that it might be this kind of thing like a death laser. Every time an animal was exposed to a mid-frequency active sonar, it would die. And of course, now we know that that's not the case at all. But it was a mess in the years following that. But the good thing was that it led to scientists, including many folks funded by the Navy, and most of my work on beaked whales is funded by the Navy, to think hard about how these animals made a living and why they responded that way. Initially, there was a lot of thought that maybe there was acoustic trauma in the ears or some direct physical result as a result of exposure to those sonars. But Walter and Zimmer, in a paper in 2007, made the point that maybe it was a behavior response, and maybe it was something akin to an anti-predator response, that somehow the beaked whales were perceiving these sonar signals as something like the signals of a predator, like a killer whale. And that's how we imagine how these animals respond to this particular type of disturbance. But it's also, that idea has grown, and a lot of what we see in beaked whale behavior, particularly in Ziphius behavior, seems like it has evolved to minimize the exposure of these animals to predators at the surface. And so I'll take that as a kind of theme as we go through talking about these animals. The other thing that the stranding event did was provide investment into new ways of studying beaked whales and other cetaceans. We take things like digital acoustic tags and CATS tags now as just a given, right? They're tools that you all use when you're studying cetaceans. But 20 years ago, those tools didn't exist. This is a picture of Mark Johnson and Peter Tyack gazing upon their invention, the digital acoustic tag. Without that tag, our understanding of almost any species that is studied widely now would be much, much, much less. And so it's remarkable to me in that just that short span of time, 2 decades, that this tool has provided so much insight into how cetaceans make a living. And we depend on that tool in addition to others, as I'll make the point. So that's 2003. By 2013, the first paper comes out looking at the response of Cuvier's beaked whales to both simulated and real sonar. And this is a paper led by Stacy DeRuiter, but you can see Peter's name there and a lot of other folks that many of you will know. And this provided the first real insight into how these animals responded when they were exposed to this type of sonar. And that response was very consistent with the idea that the animals were perceiving those signals as the signals of a predator, or the signals of something that sounded a lot like a predator. If you've looked at DTAG data before, this will look very, very familiar to you. This is the dive profile up here. So the animal's making a series of relatively shallow dives, deep for most species, and then a very deep dive. And the blue here is just indicative of the fact that the animal's echolocating, so foraging. And you can see the animal there are 3 deep dives in the record, and the animal is silent when it's not, when it's above about 500 meters. Again, consistent with this idea that the animals are cryptic. They don't want to advertise their presence to surface-dwelling predators like killer whales. Here's the sonar exposure, and you can see here that it looks like the animal changes its behavior. You would expect another deep dive here, but instead the animal stays relatively shallow. And this is fluke interval. The animal's fluking very hard, and it's swimming very hard away from the source of the sound. So the animal, again, perceives a signal that it associates perhaps as a predator. It stops foraging, it goes silent, and it swims very hard away from that sound. So again, consistent with this idea that that the animals are perceiving those sounds as the sounds of a predator. About this time, we are starting a program at the shelf break off Cape Hatteras. And Lee was part of that back when we were younger. We were working in this really amazing environment. So here's Cape Hatteras here. And you can see this is the place where the Labrador current flowing down from the Arctic collides with the Gulf Stream flowing up from the tropics. This is a major biogeographical barrier on the US East Coast. It's where subtropical species kind of collide with temperate boreal species. It's a very rich place. And this is Cape Hatteras here, and this is the shelf break. And the shelf break's about 30, 35 miles from shore. It's the narrowest part of the shelf on the US East Coast. So it's a great place to study pelagic animals because you can get out to them relatively easily. And we knew that it was a rich place because of the stranding record. We had tons of strandings of pelagic animals, including beaked whales, several species of beaked whales on Cape Hatteras. But nobody had ever worked out there before, apart from an occasional NOAA stock assessment survey. And we were interested in working out there because we were trying to solve the problem of pilot whales interacting with the pelagic longline fishery, depredating bait and catch. And so we were working on the short fin pilot whales out there. But our first kind of entree into beaked whales was from the work of Joyce, who was a PhD student in my lab. And she was looking, one of the things she looked at in her dissertation was the distribution of beaked whales along the entire US East Coast. So she analyzed passive acoustic recordings from bottom-mounted monitors from the gully up off the Scotian Shelf all the way down to Jacksonville. And this is from her 2017 paper. And the plots on the right just show you the days with acoustic presence of these different species. So bottlenose whales, sourbees, beaked whales, cuvier's beaked whales, Ziphius cavirostris, gervais, and blainvilles. And you can see up here in the gully, this is the gully, mostly bottlenose whales. That's where Hal Whitehead has his long-term study of bottlenose whales. We move down to Cape Hatteras, you can see that cuvier's, Ziphius, are found almost every day there, at least heard every day there. And really interestingly, when you just go around the corner from Hatteras to Onslow Bay, which is just mostly south of the Duke Lab here, the Ziphius kind of run out and then are replaced by gervais beaked whales. So some really interesting fine scale patterning of habitat use of these two species. At this time, we'd really never seen a live Ziphius or worked with them out there at all. About the same time, Bill McClellan and Ann Papps from UNC Wilmington were flying aerial surveys for the Navy, looking at the distribution of pelagic species in that area. And they confirmed the presence of these beaked whales that we'd heard on the bottom-mounted recorders. So the Ziphius sightings there on the transect lines are in the blue, the gervais are in the green crosses, and you can see very dense aggregation, of animals from aerial surveys. Aerial surveys are about the worst possible way to monitor beaked whales because they spend so little time at the surface. But Bill and his colleagues made the point that this is a very dense aggregation of Ziphius off Cape Hatteras. And again, you can also see that the Ziphius, when you come around the corner past Cape Hatteras, the Ziphius kind of run out. And that's consistent with what we've seen in years since. So Lee and me and others were out there playing with short fin pilot whales, trying to figure out what they were doing, how they were taking tuna and squid off pelagic longlines. And we were putting D tags on animals, looking at their behavior. Lee and Todd were out there with us getting biopsy samples so we could look at stable isotopes, and working really hard on this question of depredation. At the same time, we had Ari Friedlander and Dave Johnson out with us helping. And if you know Ari and Dave, you won't be surprised to see them like this. They're on a small boat. So we'd have multiple small boats out there working on pilot whales. We'd be working, like following a group with a D tag, and Ari and Dave would be on the other boat, and I'd turn around and look, and they would just have disappeared. They'd gone. And like, where the hell have they gone? And then hours later, they'd come back, and Ari would, yeah, we went to look for Ziphius. And so he would wander off 5 or 10 miles further offshore to look for beaked whales, not doing what he was supposed to do. If you know Ari, that's not at all surprising. But he found where the beaked whales were. And so another 5 or 10 miles past the shelf break, so about 40 miles from shore, we can regularly find Ziphius, and that's what it looks like. It's in the middle of the Gulf Stream. So imagine a river of water that's flowing at 6 knots. It's this beautiful, incredible azure color. There's nobody out there because all the charter boats work on the shelf break. So we're by ourselves, and there are tons of Ziphius out there. It's an incredible place to work. I'll just show you what it's like. I'm going to show you this little video film. This is a GoPro that Will Trophy took just to give you a sense of what it's like, 40 miles off Cape Hatteras. This is in our rib. That's Heather Foley, who worked for me for more than 10 years. He's just moved to Woods Hole. And Will's just going around the back of the boat. That's Zach, who runs our field crew. One of the reasons I wanted to show you this video is to give you these names because these people are one of the most highly skilled field crew I've ever worked with. I love them. We couldn't do this work without them. So here we are approaching a group of Ziphius, 4 animals in the group. And you can see the conditions are really beautiful, right? That's the only way we can actually work with Ziphius. You need a Beaufort, no more than a Beaufort 2. And remember, we're also 40 miles from shore in a 30-foot boat. So we have a long commute to get out there. And we get the crap kicked out of us sometimes on the way back in. But this is what it's like. So a group of 4 animals, and it turns out, and I'll show you the data that supports this in a second, we have very little time to get close to the animals because they spend so little time at the surface. So we have to be very efficient with anything we want to do. Take pictures, put tags on, collect biopsies, anything, because the animals are with us for such a short period of time at the surface before they go back again. And again, it's because they're not comfortable at the surface because that's where their predators are. There's an adult male. The adult males are very white. They're scarred. I'll talk about that in a second. The brown animals can be either females or juveniles. We're working to try and sort out which is which. We use lasers to try and estimate length of the animals. So that's a good example of a brown animal. It could be a juvenile or an adult female. And just so we started to use the techniques that had been developed by others. So this is Zach deploying a D-tag on a Ziphius. I'm driving. I've got no sound on because Zach is yelling at me because I go right by an animal and he's like swearing at me right now. And then, oh, like a miracle, another animal comes up. Hero. I did not see the second animal, but still. The point I want to make to you is that it's very, like our interactions with those animals are so brief. We spend so much time at the surface waiting for them to come up, and then we have to be incredibly efficient. And we have to spend an enormous amount of time looking for them. When we have a tag on, we can relocate them, but before we get a tag on, it's really tough. The other thing that was happening about the same time, Greg Shore and Aaron Falcone were working with Russ Andrews to develop limpet tags and deploy these limpet tags on Ziphius in Southern California. And this is the paper that they wrote that got a lot of press about their first 8 animals that they tagged. And the great thing about these tags is that they give you a longer window of time than the D tags. The D tags are wonderful because of the richness of the data, but those records only last for about a window of a 24-hour period in the life of an animal. With the satellite link dive recorders, they're very coarse, but we can get data for weeks and sometimes months. And so we adopted that strategy as well. And so we brought Daniel Webster in, who'd worked with Russ and Greg and Aaron. And Daniel now is core part of our field team, and he deploys the satellite linked dive recorders for us in Cape Hatteras. This is a pretty typical adult male. You can see how scarred the animal is. Those scars are a result of agonistic interactions with other males. I'll show you a picture in a minute, but all males have 2 tusks at the distal part of their lower jaw, and they use those tusks when they're sparring with each other. So you can see the tag there, and those tags allow us to track the movements and diving behavior of animals for weeks and months. So the first thing we wanted to do is understand whether we had a resident population or whether animals were just part of a very large a larger population that spanned a bigger geographical range. So Heather Foley did her master's thesis while she was working as a tech in my lab, and this is one of the papers that came out of her thesis. And you can see that this is 20, I think, animals, some of the first 20 animals that we've tagged more than 80 now. And the animals really are resident. Occasionally they'll go walkabout. We've had animals go as far north as Norfolk Canyon. which is off the coast of Virginia. But that's, or Delaware, that's very unusual. And typically they stay in this area of really high density. We're not quite sure what is responsible for that density, but we're thinking about it, and I'll be happy to talk about that. Jeannie Sharer, another PhD student in my lab, wrote up our first description of the diving behavior following the paper that Greg and Aaron and Russ had produced. And we see very similar patterns. If you remember that very short window of time from Aaron Falcone's, Stacy DeRuita's paper with Ziphius, same thing we see here. Long dives, deep dives, followed by a series of relatively shallow dives, usually to less than 500 meters. Jeannie is a very good analyst, so she was able to take these data, and here's a blow up of them. So those long dives are foraging dives. Those short dives, we sometimes refer to as bounce dives, but it's important to think of the entire interval between one long foraging dive and the next foraging dive as something that's very different behaviorally than the foraging dive, partially because the animals are silent when they're in those surface waters, right? So here, the animal, when it gets to below about 500 meters, will be clicking until it gets almost up to 500 meters again, and it's silent. So when the animal's up here, even though it's diving to 200 or 300 meters, it's not making any noise whatsoever. So Jeannie's analysis was able to separate the shallow dives from the deep dives. And we know from the DTAG records that those deep dives appear to be foraging dives. The animals are echolocating. Occasionally we can get echoes back from prey. And so we use these cutoffs. Anything longer than 33 minutes, we take to be a foraging dive. Anything shorter than that as in part of an inter-dive interval, into deep dive interval. And all the deep dives are basically greater than 800 meters. This shows you, these are the 10 animals that Jeannie analyzed, the depth, frequency, histograms of the depth, means in red, medians in blue, and then the durations of those dives. So the median depth of a foraging dive for a Ziphius is 1,500 meters. And that's just kind of a remarkable thing. We kind of get inured to that, right? So I always tell my students, just imagine you're going to run 3 kilometers and you take a breath when you start and you don't breathe again until you've gone 3 kilometers. And that's what a beaked whale does every time it wants to forage. The average duration of those dives is 59 minutes, an hour, holding your breath for an hour while you're getting lunch. So it's kind of just remarkable. It also explains why they're so hard to study, right? If you come upon a group of beaked whales just as they're going down for a foraging dive, you have to wait at the surface for an hour for them to come back. And when they're at the surface, and we knew this qualitatively, but Jeannie quantified it for us, they're at the surface for, on average, 2.1 minutes in each time they come up to breathe. So you're waiting for an hour for them to come up, and then you have 2.1 minutes to go and do everything you want to do. The terminal dive is the dive just before they go on a foraging dive, a deep dive, and you can see that they spend slightly more time at the surface then because they're storing oxygen before they go down. This is the first dive after a deep dive, and this is an intermediate dive. Occasionally, and you can see how few records there are here, occasionally they'll come to the surface and take a single breath and then stay at the surface. We don't know what these are. They're not very common. We always hope that when we come behind a group, we'll get one of these times where you'll have 10 or 15 minutes from the animal. I've only ever had that happen to me once. And again, we really don't understand what this behavior is. It's very different from the typical diving pattern. So we had started to accumulate all this baseline information. We'd been funded by the Navy, and then we pitched the idea to the Navy of doing a controlled exposure experiment off Cape Hatteras to really understand the response of these animals to sonar. We'd had the early work done by Stacy and Peter and John and Brandon, and there'd been all the other work done in SoCal led by Brandon. But we thought this was a really interesting opportunity to understand how these animals would respond using all the tools we developed in this population that we had really good access to. So this is the project that consumes my time in the summer now. It's the most complicated project I've ever been part of. It's great because we learn a lot about beaked whales. I'll just say a couple of things about the project because it frames interesting things that I think we are learning about beaked whales that have nothing to do with sonar. I'm not going to talk about the results of sonar. I'd be happy to talk about that afterwards, but I'm not going to talk about it very much. So this is an adult male with a D tag. That's our research vessel that we use for overnight tracking the shearwater in the back. It's a great platform for doing this stuff. So the objectives of our project, and again, this is just framing some of the questions I want to focus on after this. We really want to understand how Ziphius respond to Navy mid-frequency active sonar tactical signals. And these are the factors we're interested in: sound level, like how loud the signals are, how close the signals are, and whether animals in different behavioral states, whether they're on a foraging dive, matters in how they respond versus whether they're in that into deep dive interval. We want to know whether they move away, have changed the behavioral state, and also because we had started to get some really interesting inference into their social behavior, how social behavior, how groupings or what the animals were doing with each other would mediate their response. Cape Hatteras is a good place to work because it's not on a Navy range. Most of the work that's been done on Ziphius in the US has been done in SoCal, which is a range, so they're exposed to sonar all the time. And then we wanted to use, especially the limpet tags, to get a longer picture before and after the exposure. The problem with the D tags is you've got this little tiny narrow period of time. And so you only get the very immediate response and you get very little baseline before. And with the satellite link dive recorders, we can get weeks before and weeks afterwards. So this is how we go about doing it. We, prior to an event, will deploy a lot of tags, a lot of limpet tags. And then we conduct a controlled exposure experiment in which we expose a focal animal and the other tagged animals to either operational sonar from a Navy vessel or simulated sonar from a scaled source. We attempt to have a D tag and multiple splash tags in the same group, so we can get both the fine scale response and the coarser response. And then we also pay particular attention to what happens to the group composition, whether the group splinters, whether the animals stay together. One of the issues we had right away when we first started deploying the limpet tags was that the data were very gappy. So you can transmit to Argos 32 bits. And so that's a really narrow pipe to transmit up to the satellite. The animals are only at the surface for 2.1 minutes at a time. So often you'd get part of a message up to the satellite, and you wouldn't get all of it. So we had all these long gaps in the record from the satellite link dive recorders when we didn't know what the animals were doing. And from the Navy's perspective, from the controlled exposure experiment perspective, we couldn't possibly have that and have the potential like bringing a Navy destroyer in and have the focal animal have a gap in the record so we didn't know what it was going to do, right? That would be the worst possible outcome. So here's Nicola Quick, who is a research scientist in my lab. She's just taken a position as a lecturer at the University of Plymouth in the UK. And she and Will Chofie worked really hard to modify the settings of the tags so that we could get complete records. And they did an incredibly beautiful job. So now we can get full records without any gaps for periods. We've done it for 14 days, and last year we extended that to 21 days. So we get a continuous record of the diving behavior for a 3-week period for every animal. We do that by sampling depth every 5 minutes. So think about the D-tag record, how finely, how beautifully rich that record is. This is a very coarse record, but it's long. To maximize the amount of data we get back, we also use an Argos receiver that's known as a goniometer. And we spend time in the field actually harvesting data, intercepting the signals that are going up to the satellite so that we can make sure we don't have any gaps. That's proven to be a really important part of our field component. It also allows us to use the satellite tags as beacons to be able to find animals in the field. We still regularly break tags. And because the animals are diving so deeply, we have had animals that have gone past 3,000 meters, and the splash tags are just not built for that. So we are continuously having animals break tags. We've worked with wildlife computers to try and improve that, but we look at the pressure sensors on those tags all the time, and we lose tags regularly because the animals break them. So just to show you, these are two animals. There are two dive records here. The black line is a D tag. So we're getting that continuous record, well, almost continuous record of depth. And then in the same group is an animal with a splash tag that we've programmed. And those red dots and the blue error bars are what the animal's doing. The animals dive, when they dive, they're completely synchronous. I'll show you some examples of that. So you can see we can use the D tag record as a proxy to see how well the splash tag is doing. And you can see it's pretty good. Because of the weird way that the algorithm actually samples depth, we get more error as you go down, but you can see it's still a pretty faithful record of the depth on the splash tag. So we're really happy with this. We can use the splash tags as long data records to infer what the animals are doing. I think this is the last thing I'm going to say about the Navy. The thing that I'm proud of, we've done 8 controlled exposure experiments with actual Navy destroyers. These are vessels that are home ported in Norfolk. These are multi-billion-dollar ships with crew of 300, and we get an hour of their time and to come and play back sonar signals in a controlled way. We do real-time acoustic propagation modeling so that we are giving a dose of sound to an animal that's loud enough for the animal to hear and respond to, but not too loud to overwhelm it. And the most complicated thing, working with beaked whales is complicated. The most complicated thing is communicating with the Navy. So we can be out there and the Navy destroyer might be, they're usually, when we're doing a CE, they're usually about 25 or 30 kilometers away. We could conceivably just talk to them on the radio, but they won't answer. So what we have to do is get on the satellite phone, call somebody on shore, who calls somebody in Norfolk, who walks into the secure room, who then communicates to the Navy vessel. And you can imagine that that never gets screwed up at all, right? So that's the most complicated thing, but we've had some really good results, and the first paper is just about ready to go, and the second paper not far from it. So, and I'm happy to talk about that. During this time, we spent a lot of time doing this. That's me in the pulpit of the barber waiting for an animal to come up. And so we sit around and we talk about beaked whales and we have questions. The most important one we spend a lot of time talking about, like, are these really mammals, or are they creatures from another universe who've come here to taunt us? And our research group is split on the answer to that question. But we spend a lot of time, imagine like 59 minutes waiting for an animal to come up. You can only talk about like what you're gonna have for dinner so much. And so eventually the conversation gets around to beaked whales. I'm gonna talk about 2 of the remaining 4 questions. How do they stay submerged for such long periods? And then what are they doing socially? Because if you go back to the literature, the old stranding days, people view these animals as solitary, but they're not. They're really social animals. They have tusks, they fight with each other, they occur in groups. And so we're starting to get really interesting insights into their patterns of social behavior. We are very interested in what they're eating down there, 1,500 meters, and I'm happy to talk about that afterwards. And I might touch on this last one, that, like, does the anti-predator hypothesis hold for Ziphius? We think it does, and we're pitching an idea to ONR right now to actually test that, but yeah. So how do they stay down for so long? I don't have the answer to that, but I'm going to get you to think about it with me. So Nick also published this paper a couple of years ago. It's a short paper in JEB in which we wanted to know what's the diving capability of the species? Like, how long can they stay submerged without pushing themselves? And part of that is out of pure interest, but part of that is also how we should interpret the response of these animals to the sonar signals. Like, do they stay down way beyond the time that their oxygen stores allow them to do so, so they go anaerobic? So we tried to think about how to do that, and we went back to the literature developed by Jerry Kriemann and came up with this idea of a behavioral aerobic dive limit. And what Kriemann found with his Weddell seals was that 95% of the dives that a Weddell seal would make would be within its aerobic dive limit, would be long enough so that the animal still had oxygen to use. So the animal didn't have to undergo anaerobic metabolism. Because if you undergo anaerobic metabolism, you have to recover from that metabolism, and that's lost time. And so the idea is, if you look to see where 95% of the dives are, and that should, if what Kriemann found for Weddell seals holds for Ziphius, that should tell you what the oxygen stores of the animals are. So Nick looked at a much larger data set, more than 20 animals. And so she had more than 3,600 foraging dives to look at. So she looked at the distribution of all these dives to see what it could tell us about how long they could stay submerged and still remain aerobic. So this is the histogram of dive duration. Median duration was exactly the same in this data set as it was from the one that Jeannie analyzed, 59 minutes. So on average, the animals are staying submerged for about an hour. The 95th percentile is at about 78 minutes, suggesting that the animals of average size can stay submerged for about an hour and 18 minutes before they run out of oxygen. But there you can see that tail extending way off to the right. So animals will sometimes make very, very, very long dives. They don't. They really do not. So the plot on the left is a scatter plot of dive duration versus the next into deep dive interval. So how long the animals spend at the surface after a dive of a particular period. And you can see that there's a positive relationship there, right? Which makes sense. So the longer the dive, the longer the animals will spend in that into deep dive interval, coming to the surface, but then going back down again to a couple hundred meters. So they're taking more breaths because they have to recover. The weird thing that we cannot explain, nor can anybody that we know who we've talked to, and we've talked to all the respiratory physiologists we can find, when you look at the top 5%, the longest 5% of the dives, and that's what's in plot B here, that relationship breaks down completely. So that means when an animal makes a really, really, really long dive, which we expect to exceed the animal's aerobic dive limit, which means that it's utilizing anaerobic metabolism, it should have to recover from that, and it doesn't. It's like you running a marathon and then saying, okay, I'm just going to run another marathon now because I'm not tired, right? Mammals don't work like that. And Ziphius shouldn't be able to do that. So we are puzzled as to how they can do that. The potential explanations are either that they can reduce the metabolism in such a way that they are using very, very little oxygen. I mean, way less than we would expect them to be using at a resting metabolic rate, which seems unlikely. Or that they have some novel process to either use lactic acid in a pathway that we don't know about, or to somehow store lactic acid and then deal with it later. Neither of those is known in the mammalian physiology literature, but they're doing something that no other mammal does. And we would love to know what that is, because it would have, I think, probably pretty profound impact into how we view how mammalian physiology has evolved. Paper got a lot of press, not because of all the beautiful work we did, but because of one record, right? And we kind of knew that. So one beaked whale stays submerged for 3 hours and 42 minutes. Like that should not happen, right? And we looked and looked and looked, and it's not an error. We looked every way we possibly could. And there are other ones that are nearly as long as that. This one didn't make it into the analysis because we censored all the data so that after an exposure to sonar, we didn't include any of that data. This was about 2 weeks after exposure to sonar. So it's inconceivable to us that exposure 2 weeks earlier could have affected it. But somehow this beaked whale was able to stay down for 3 hours and 42 minutes. It did spend a lot of time at the surface after that. But imagine staying submerged, holding your breath for 3 hours, 42 minutes while you're doing something, right? It's amazing. Okay, let's talk about social interactions. So this is an adult male surfacing. You can see the tusks there, the two tusks. The tusks have barnacles growing on them. We have a very eccentric barnacle biologist at the Marine Lab, known to those of us, Dr. Dan. And so I've asked him, like, how the hell do these barnacles live as they're being cycled from the surface down to 1,500 meters and then up and then down? We don't know that either. So they're pretty interesting barnacles. But this is an adult male. You can see the tusks and see how white the animal is, and you can see some of the scars there. One of the things we've been really interested in is like splits and joins, right? So when animals come to a group and when they leave a group, and again, that's helping us to interpret what happens when the animals are exposed to sonar. So this work's done by Danielle Waples, who does all the photo ID in my lab, and she spends most of her life looking at pictures of Ziphius. And she does a great job. She's really detailed. So very appreciative for all the hard work she does. So here's and a group of animals on 9th of August, 2020. There are 5 animals in the group, 5 animals with tags. They're all tagged. We tagged one and then we went back and found it. Group again tagged it and so forth. And 4 of them are adult males. We know that from the pigmentation. The other animal is either a juvenile or an adult female. We find them 10 days later. 108 and 71 are still together, but now there are 3 new individuals with them, including another adult male. Here's 108 with the satellite tag. The next day, we find them again. 108 is still together with 71, 84, 135, 136 are all together. They've been joined by another non-adult male. And here's 71, here's his buddy with he's been both satellite tagged and detagged. And so just to the point, like these are animals that exhibit fission fusion, social organization, just like bottlenose dolphins do. They have very rich social lives. They don't spend all their time together with each other. And we wonder how those social groupings mediate their response to disturbance like sonar. But we don't know yet how that actually works. This is from Will Chofee, another PhD student in my lab who finished a couple of years ago. He did some really beautiful work looking at the social organization and social behavior. And Will hypothesized that because these males have tusks, because they fight with each other, that there would be no stability in groups with more than one mature male. And he found exactly the opposite thing. So here is a group, 2 adult males tagged in the same group. And you can see this beautiful synchrony of diving. The animals are just perfectly synced up, right? Here we have 2 animals of different age and sex classes. It's an adult male and a brown animal. We don't know if it's a mature female or juvenile. And you can see it's not nearly so synced. So the question is, what's going on with this synchrony with the adult males? So there are a couple of hypotheses. Either the males are working together in coalitions, like we see in bottlenose dolphins, or that synchrony is part of a display. There are some examples in the literature, like some cervids, some deer, the kind of agonistic display between males involves things like parallel walking. And so maybe it's part of an agonistic display in which the males are jockeying for dominance with each other. We don't know yet, but we're working on it. It's really, I mean, again, the richness of their social lives is pretty impressive. I have an undergraduate student right now, an REU student, who's looking at animals that we have pictures on successive surfacings, like deep dives and then shallow dives, to see when scars are acquired. So if you think about this dive profile, right? So the animals here are foraging. We don't expect them to be socializing when they're foraging. And that means they have to be, if we're correct, we might be wrong. If they're correct, then they have to be socializing during this period when they're silent, which is kind of interesting, right? So it's strange. So, but we want to know whether animals, like if we see an animal here, does it acquire scars between here, new scars between here and here? Or if we see it here and then see it again here, does it acquire scars during the deep dive? And we have a lot of photographs now that she's working through. So it's going to be interesting to see how that turns out. In addition, we just got to figure out who these brown animals are. And so this is an animal wearing a satellite tag, and there's a biopsy dart. So we're getting a biopsy sample from it. So we're working really hard to, especially with the brown animals, to get sex, so that we'll be able to better interpret the social groupings that we see. And it's hard, right? Because we have 2.1 minutes to work with an animal. So you have your choice. You can put a tag on, you can take photos, you can get a biopsy sample. You can't do all 3 of those things. You can do 2 of them. And so usually we're putting tags on and getting photos. And so the biopsy sampling has kind of trailed behind a little bit, but we're starting to work to catch up now. And this summer, we're going to have a big push to get biopsy samples so we can get more sex information. We're also working with Greg and Aaron, who are leading an ONR project. There are 4 groups of researchers that are studying long-term projects on Ziphius. Greg and Aaron in Southern California, us in Hatteras, group in Guadalupe in Mexico, and a group in the Mediterranean. And the ONR project that we're working on now, we're using exactly the same scoring system for scarring to assign age and sex. And we're kind of pooling our information in all our biopsy samples so that we're hopefully going to be able to make some cross-population comparisons about age, structure, and social groupings. One of the really weird things is the incredibly low fecundity of this species. We very rarely see calves. We see them, but and that's true for all 4 populations. It looks like the fecundity rate of this species is really, really low. And we have no idea how long they live. It could be that they live for a very long time, and so they have a very slow life history. But it's interesting. Okay, I'm just about to finish up. I want to make sure we have time for questions. I'm just going to touch on this question of the anti-predator hypothesis. This is a paper came out last year, I think, in PNAS, and Patrick Miller is the lead author on it. And these are his co-authors are all part of what's known as the 4S project, and they've been doing controlled exposure experiments in Europe and some really beautiful work. And Patrick brought this kind of all of the work together to assess whether that anti-predator hypothesis holds when we look at the response of different cetaceans to sonar signals. They don't look at Ziphius. They looked at bottlenose whales in the subarctic and the Northeast Atlantic. And certainly for those animals, the evidence is consistent with the hypothesis that these animals respond to those sonar signals like they would respond to predators. And if we listen to the signals themselves, that we think about sonar, you're probably thinking about a ping, 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 like in Red October. But the mid-frequency active sonar signals are not like that. They're frequency modulated sweeps. And so to our ear, they do sound quite a lot like the sounds of killer whales. The response we see in our Ziphius from all the playbacks we've done now is entirely consistent with the anti-predator hypothesis. That our animals, when they hear sonar, whether it's simulated sonar or the sonar from real sonar from ships, they go quiet, they orient. One of the things they do that's really cool, we see from the D tags, they spin. As soon as they start to hear that, they'll spin. They go silent and they spin. And what we think they're doing is they're listening, trying to get a sense of where the sound is coming from, and then they move away from it. And if the sound stops, they move away really, really fast. Like if they can still hear the sound, they'll move away at a pretty good clip. But if the signal stops, then they run really hard. And that's consistent if you think about killer whales preying on marine mammals. Typically when killer whales prey, they go silent, right? Because they don't want to advertise themselves. So it's all consistent with this idea that beaked whales perceive those signals as the signals as similar to the signals of their predators. And we're going to make a pitch to Mike Weiss at ONR this year because we have this big infrastructure now, because we're putting tags out, we're playing back sonar signals. Want to add another layer of complexity, and to some of the same animals that we're going to play back sonar to, real sonar to, we want to play back killer whale signals as well, just to make sure that the response is the same. And we can think about the order in which we do that, but because we have these long duration tags, I think we can do it. So, okay. So I'm going to end there. Good, I've got time. I just want to, there's a huge number of people to thank. I hope I've named all the people in my group. And again, my field team, Zach Swain, Daniel Webster, Heather Foley, are some of the most gifted scientists I've ever worked with. And we couldn't do this if not for their skill and their dedication. But a whole huge number of people have made this possible. And I'm really enormously grateful to work with all of them. And with that, I'd be happy to take any questions.
All right. So just like we normally do, we're going to bounce back and forth. Are there any questions online, Roseanne? Okay, if you have questions for those folks on our line, put them into the chat and we'll work through them. I see questions in the room.
Cool talk, Andy. Do you have a lot of length data from those paired lasers?
We have some. They don't work nearly as well as they should. I like showing it because it's really cool. But it's really bright out there. It works on some of the animals, but not some. We have some, but not a lot. Okay. And the relationships we have, the morphometric relationships we have, like you anchor it with the length of the dorsal, the width of the dorsal fin. There's crap morphometric data out there for the species. We have we do it really well with pilot whales. It's not very good with this. Any repeated measurements of the same individuals? Only, I think, one or two, enough to convince us that it's not very accurate.
Cool.
But it looks good on the video.
Great talk, Andy. That was awesome. How often do you guys see killer whales out there?
Right. We never see them in the summer, but they're off Cape Hatteras every winter. And they're known by the charter fleet because they take giant bluefin tuna off troll gear. So if you Google killer whales, bluefin tuna, Cape Hatteras, you'll see video of a bunch of fishermen who just lost a big bluefin to a killer whale. They don't care so much because they've already got a big bluefin in the boat. And they're like, whoa, man, look at that. That'd been the first bluefin tuna, they would have been pissed. But so they're there in the wintertime. They're not there in the summertime. But we also know that the density of killer whales in the Atlantic is, it's just a remnant from what it used to be because they were largely extirpated. They were viewed as pests in the 50s, 60s, 40s, 50s, 60s, and 70s. And so they were almost extirpated. But they're there. There are just not very many of them. But think about the landscape of fear, right? If you're a Ziphius, you're not take a risk, right? If I said there was a cougar wandering around here in the island, your chance of encountering it might be low, but you'd probably stay here, right? So I think it's pretty hardwired into them. Yeah.
And just to follow up, you'd expect killer whales out there, all the fish-eating ones, to be separate from the mammalian.
Yeah, the Atlantic, we don't know. So we know very little about the foraging ecology of killer whales in the Atlantic. There's some work done in the Caribbean, in which they certainly take marine mammals down there. The fact that the killer whales take bluefin tuna off troll gear suggests to me that they're certainly capable of taking Ziphius. You can also Google Ziphius and killer whales in Australia, and there's some really gruesome video of killer whales taking down multiple Ziphius there. So it happens. Yeah.
Questions online? Not yet. Okay, questions in the room. I'm going to go up there because you kind of have it in with this particular speaker.
I have two questions.
Yep.
The first is in regard to the short intervals of dives.
Yep.
Are those likely due to like predation risk? And that's the reason they dive down to 500 meters or so?
Yeah, it's a really good question. So initially, when Stacy and Peter first got those initial dive records from the species, they hypothesized that the shallow dives, what we refer to as bounce dives sometimes, were used for the regulation of respiratory gases. That somehow, I mean, think about what these animals are doing from a physiological perspective. And they thought maybe there was something about the animals needing to deal with their nitrogen in the bloodstream. We don't think that's true. If you think about an animal say an animal dives down to 1,500 meters, right? And it's trying to conserve oxygen as much as it can, or it's evolved to do that. So that means you're going to shut down oxygen flow to pretty much everything except for your brain and your heart, your uterus if you're pregnant. And that means that you're not going to do a whole bunch of things that we're all doing right now, like digesting, right? They'd stop digesting. Will Chofie says they stop thinking. He says that that's how they do it. They shut off blood to their brain. They don't think very much. He's kidding. But so that means they have to do all those other life functions at some other point, right? So we think that those shallow dives are when they do everything else. They digest, they socialize, but they don't want to do it at the surface because at the surface, they're at risk. So they go down a couple hundred meters and they shut up, they're quiet. But then that's like, how do you socialize if you're not making sounds as a mammal? And so it just gets interesting very quickly.
My second question is involving your course trackers. You mentioned that a lot of the whales will often break them if they go too deep. Is it difficult to engineer a tracker that survives deeper than 3,000 meters?
It's not difficult. So what happens? So the tags have a pressure sensor on them, and then the pressure sensor is monitoring pressure and then at some interval, it's recording that, and then it's going to put that into the algorithm that decides what's going to be sent up to the satellite. The pressure sensor breaks because the tag itself is not housed sufficiently to withstand the depths at which animals go to. Certainly not, they're not, I can't remember what they're rated to. They're not rated to 3 kilometers. What we do is we look at when the animal comes to the surface and that the saltwater switch goes off, indicating to us that the animal's actually at the surface. The pressure sensor should read 0. And so we get little snippets of those data to make sure that that happens. And what we see when the tag breaks, when the pressure sensor breaks, is we start to see drift. We know the animal's at the surface, but the pressure sensor says it's the 100 meters. And then we throw the data out after that. So we've had many conversations with the engineers at Wildlife Computers, pleading, begging, cajoling them to build a tougher tag. But I think for them, because they're a small company and there are only a few of us who are putting these tags on really deep diving whales, it's not worth their while economically. But we need a better tag.
Thank you.
You're welcome. Good, great questions.
Any other questions in the room? All right.
Great talk, Andy. Thank
Thanks,
you.
Jim.
With a nod to Lee, I was wondering if you ever sample feces for stress hormones, reproductive hormones, or anything of that sort.
We have never seen Ziphius poop, ever. We have looked for it. We see pilot whale poop and we get all excited about it. And so I think, again, so we think that, they come up, they're at the surface for just a couple of minutes. You would think that would be a good time to poop, but they don't. I think it's at some other point in those shallow dives, they're pooping. So I cannot tell you how excited we would be if we saw it, but we've never seen it.
So what the heck are they eating anyways?
I was hoping somebody's going to ask me that. So what we do know is that they are foraging basically on the seafloor. We often get echoes back from the seafloor from the D tags. We know from the depth records that they're going basically to the bottom. In several places of the world, there are records of what are referred to as foraging pits that look like when beaked whales are going down and actually grabbing something like either in the sediment or close to the sediment. We don't know what they're eating, but there have been several NOAA deep ROV explorations of the system that we're working in. So there are a lot of pretty large prey down there, things like rat tails, big fish. And here's the cool thing. This is why I wanted somebody to thank you for asking this question. Imagine that deep sea ecosystem, right? So these fish have for many, many, many tens of millions of years, maybe longer, evolved down there. Light is really important to them, so there's the evolution of light production and really good vision to take advantage of what light's down there. They were no predator ever came down there using sound to find prey. And that's what Ziphius does, right? They don't care about the light so much, but they can use sound, and none of those prey are defended against it. So what we think is that the cost is, and it's the same thing for, I would argue for elephant seals. There's a big cost in terms of the commute to get down there. Once you get down there, there's a lot to eat, and that stuff is not hard to catch. They're suction feeders. They don't have, males have the 2 tusks, females have no erupted teeth. So they just go around like slurping up dinner. You have to pay the cost to get down there. And so thinking about the evolution of like predator prey in the deep sea, we have a biologist at Duke, Sonke Janssen, who's talked about this a lot, but he thinks about how light is used. And these predators coming down don't care about light, they use sound. And so to your question, we think pretty large bathypelagic fish for the most part. There are stomach contents, but the stomach contents are from stranded animals that are sick and have died, and they're not really, I don't think they tell us very much.
All right, we're gonna do one more question here. Hang on.
Okay.
Hi,
Last
Andy.
question, but it'd be a good one.
Oh
No
my
pressure.
God, it's a lot of pressure. I was going to do the same question actually, but I was just wondering if you had the opportunity to collect tissues of the animals that you mentioned, like the shallow water divers and the deep water divers, to see maybe if they're consuming different prey sources.
So we have biopsy samples, and we can do things like stable isotope analysis. We could do fatty acid analysis, but we have really no library of prey to compare them against. So what we dream of is to have a camera that we could actually see what they're doing down there. The other thing what we I think hopefully we're just about to get funded for, again by ONR, is with 2 other groups looking at environmental DNA. So what we're going to do is to have basically go to the footprint of a beaked whale on a foraging dive, and then sample the water down near the sea floor and then see what we get from environmental DNA to see exactly what might be down there. So that's hopefully we'll start doing that. So we're trying to think about these indirect ways to try and understand more about what they're doing in terms of foraging, but it's hard. I mean, working at 1500 meters it's dark, it's a lot of pressure, it's cold, it's weird. So
I
yeah, very good question.
was going to say we could end on the weird note,
Yeah.
and just say one more time, thank you so very, very much.
Thank you guys for coming.
All right. And hopefully we'll see you next week. Same time, same place. Thanks, everyone. Okay, folks on the line, we're going to end the presentation now. Thanks for joining us.